Hello students, welcome back. So we will be talking about the transcription in the eukaryotes. Generally, we know that transcription nothing but it is a process of transfer of information from the DNA to mRNA. Here we know that the enzyme which is very important for transcribing the mRNA is nothing but we know that it is depending on the DNA strand. Okay, that is why we call it as an DNA dependent also. And which strand is preparing? The mRNA strand is preparing. So we call this as an RNA polymerase enzyme. So RNA polymerase enzyme is very important for transcribing the mRNA strand. So it is depending on the DNA strand and the mRNA is being prepared. So we call this enzyme as a DNA dependent, and also we call it as an RNA polymerase enzyme. Now let's have the three more important functions of the types of the RNA polymerase enzymes. RNA polymerase enzyme, the first one is polymerase 1, which transcribes the ribosomal RNA also, and it is of 28 years and also 18 years and also 5.8. What is meant by S? S stands for it is a Swedberg constant, and that it was a scientist who has given this. We call it as a Swedberg, means sedimentation coefficient. If a cell is there, how much amount of the ribosome they are occupying in the sedimented in the form of sedimentation in the cell, we consider sedimentation coefficient also. Now RNA polymerase 2, it is transcribes the precursor of the mRNA. So main our mRNA which we are talking, that is a HN RNA also called, or heterogeneous nuclear RNA also we can call it. Why it is called heterogeneous? Nothing but it is showing the exons and also introns conditions. Then let's talk about this later guys. Now RNA polymerase 3, which transcribes the tRNA, that is transfer RNA also, that is 5 years rRNA, and also it transfers the snRNA. snRNA stands for small nuclear RNA. Now these are all of the different types of the RNAs. For example, rRNA, mRNA, and also tRNA. You just remember RMT, that means RNA polymerase 1, 2, 3 are synthesizing these amount of the RNAs. At the same time, they are also synthesizing ribosomes. Now let's back to this class, that is transcription in the eukaryotes we are talking. Eukaryotes, you generally know that they are well developed structure. They are having nucleus and also nuclear membrane, and they are also having the endoplasmic reticulum. Now complete transcription, it is taking place in the nucleus. So in the nucleus, what happens? The DNA strand is the one which is having these directions, and in the middle of this, we know that in the previous class we studied that there is a promoter site also, structural genes also, and terminator regions also are present here, and also it is having a tatat, that is an indication, that is right, tatat box also it will be present. Now in this strand only, the RNA polymerase, that is DNA dependent, and RNA polymerase is the one which is coming and attaching. So the newly synthesized strand, as the same we follow in the prokaryotes also, 5th primer to 3rd primer direction it is coming out. Now 5th primer to 3rd primer newly synthesized strand has been come out. Now the interesting point here is, when it come out, it is not readily synthesizing the proteins immediately. Why? Because it is forming an hnRNA. What is hnRNA? We studied that heterogeneous, right? Heterogeneous nuclear. As I told you, heterogeneous nuclear means it contains the introns also, it contains the exons also. Now introns are considered as a non-coding sequences, and exon is considered as an coding sequences. That means when any protein should to be prepared, it has to take the information from the exon and it is not from the intron. Then what happens? So they has to remove the introns. So for this reason only they are getting the energy to balance this chain. What kind of a energy they are getting is nothing but capping they are making, means one cap like structure is making, that is 5-methyl guanosine triphosphate. So methyl guanosine triphosphate is considered as a cap. Okay, and in the tail region also, there has to be maintained So that in the tail region also polyadenylate, nothing but poly A tail will be added here. Adenylate will be added. Almost 300, 200 to 300 are added at the third primer end in the template independent manner. So the fully processed hnRNA is now called as an mRNA. It is transported out of the nucleus for translation. Now what happens here is, before the translation process, methyl, guanosine, triphosphate and adenylin is present. Now what are these? These are considered as an spliceosomes, or also called as an splicing. Splicing means removal of introns. These are the introns. So what they are removing, who are removing, nothing but we generally call as a snubs. Snubs means snRNA plus proteins. snRNA plus proteins. So they are the ones which are having a combination and they follow the rule. That rule we call it as an GU to AG rule. So it is a fifth primer and also they are identifying at the third primer end, and they will be following this rule. This is fifth primer end, and this is the third primer end regions. They will be identifying in these regions only, and they are the ones which are cutting this particular type of the introns, and later on they are removed out. Now this strand is having only exons, and you can get the doubt, when they get cut, then they are connecting, who is helping for that? Here comes as a DNA ligase. DNA ligase is an enzyme which connects one exon, because here gaps has come, no, because introns has been removed out. Here only exons are present, so they will be like a pieces type. So ligase is the one which is immediately coming and attaching this the strands. Now it is a fully matured mRNA strand. Now this mRNA strand will come out of the nucleus and will attach to the endoplasmic reticulum, which are having the ribosomes, and then it will play an important role for the protein synthesis. That's why we consider that the ribosomes are helping for the protein synthesis. Thank you, students. I hope you understood.